Here we are, ladies and gentlemen. We got our last video of this series talking about the immune system. So we talked about the lymphatic system, the actual organs of the immune system, I guess you could say. Technically, they're different systems. So the lymphatic system, the sort of drainage, the transport of various cells and things like that we talked about. The immune system. Now, with the immune system, I don't know if I mentioned that in the last video, but there are some organs that are involved in that. Your thymus, your bone marrow, places like that, as well as your integumentary system, and some other organ systems like that are sort of responsible for that. This video, this time around, what we're going to do is we're going to talk about the specific or the adaptive immunity. So these are the real heavy hitters as far as immunity is concerned. The specialists that I mentioned in the last video. So let's get into this, and we're going to start off by talking about lymphocytes. Now, I mentioned those at the very beginning of all of this stuff when we're talking about the lymphatic system. And so these are our B cells, our T cells, which I haven't talked about yet, and our NK cells. Now, our NK cells, we remember from the last video, those are our natural killer cells, right? Those are our surveillance cells, the watchdogs, right, that are patrolling around looking for cells that don't belong, basically. So as far as the distribution of these cells, different tissues maintain different populations of T cells and B cells without the body. They tend to be distributed throughout the whole body. They make their way through the bloodstream, or they're carried through lymphatic vessels. They, as cells go, their lifespan is pretty long. They can last for a long time. T cells and B cells specifically migrate throughout the body. They're found pretty prevalently in the bloodstream, for example, to defend peripheral tissues, especially in the case of injury, of signs of infection, and things like that. And they maintain their ability to divide. A lot of cells, once they get to their final adult form, I guess you could say it, they're not able to divide anymore. You can change the size of them, but you can't necessarily get more of them. You know, some cells like skin cells will continue to divide. Nerve cells stop once they become adults. They don't. T cells and B cells can continue to divide, and that's really important. We'll get into some of the functions of what's actually going on with these, but it's really important that these cells can still divide throughout their life and through your life in order to fulfill their particular functions within acquired immunity. So, different types of T cells. There are a number of them. We've got cytotoxic T cells. These are killer cells, basically. They will attack physically and chemistry. Helper T cells will coordinate responses of different cells, T cells and B cells. You've got regulatory T cells that moderate response. They either accelerate or they suppress inflammation. And then memory T cells are the ones that respond to previously encountered antigens. Memory T cells are the ones that we're concerned about when we're dealing with vaccines and stuff like that. These are the cells that we're activating and actively cultivating. B cells are the other type of lymphocytes. 10 to 15% of our circulating lymphocytes are B cells, which will differentiate into plasma cells. These are the ones that are responsible for producing the antibodies that you may have heard a lot about, developing antibodies for different diseases and stuff like that. And then finally, NK cells make up the remaining 5 to 10% of the circulating lymphocytes. These are our surveillance cells, our watchdogs, right? All right, here's a chart that is showing us a lot of the stuff that I had talked about. Our T cells, most of the lymphocytes circulating in your body are your T cells. The cytotoxic T cells, which are the killer cells, are going to operate kind of similarly to the NK cells. Helper T cells stimulate the activation of cells. Regulatory cells will moderate their activity, speed it up or slow it down. Memory T cells remember that we have actually come in contact with different pathogens and stuff like that. Here's our B cells that will turn into plasma cells. Those are our antibody factories, and then our surveillance system, which is the NK cells. I would recommend taking this picture that's in the, actually I think it's this direction. Hopefully I got that right. Take it, put it in your notes. Okay, this is a really good chart that sort of organizes what's going on with the different lymphocytes and stuff like that. Okay, so within adaptive immunity, we've got two different types of adaptive immunity. The first one is cell-mediated immunity. Now, we see this one primarily with the cytotoxic T cells, and what this does is the immune response is dictated by the presence of abnormal cells and pathogens within cells. So our cells, if they get infected with something like a virus, for example, or something like that, they send out really subtle chemical signals, more antigens, right? Those signal flags. And these cytotoxic T cells look for those cells and destroy them. And so in order for this immune response to kick in, there has to be a certain number of infected or abnormal cells present. That's why it's cell-mediated, okay? You've also got antibody mediated. Now, you may also hear humoral immunity, or I think we may talk about that a little bit later. I don't know. We'll see. But these are primarily driven by your B cells. Remember, those are the ones that differentiate into the plasma cells. And they are pumping out these antibodies. The antibodies, we're going to get into it in a little bit greater detail, are basically special kinds of proteins that recognize specific pathogens. And so your immune response is going to be determined by the presence of antibodies and the presence of the antigens, the pathogens that have the particular antigens that will bond with those antibodies. Here is another diagram sort of showing this. We've got immune surveillance, which is looking at viral infections, looking at cancer cells that will break open these cells. We've got antibody mediated, producing those antibodies. Now, spoiler alert, in order to have the antibody mediated response, you had to have come in contact with the pathogen before in order to create the memory T cells that stimulate the B cells that produce the particular antibodies. And then your cell-mediated immunity with infected or foreign cells, abnormal cells, that it acts similarly to the NK cells as far as finding particular cells and destroying them. Now, we were looking at this and like the T cells, there's way more T cells than there are NK cells. So there is a little bit of overlap because of the functions, but there's also some differences in which ones we see based on the percentages of cells that are floating around in our bodies. Okay, so with adaptive immunity, remember, adaptive immunity is immunity in response to an initial exposure to a pathogen, okay? And so typically how it goes is your body gets exposed to an antigen or some sort of pathogen, right? It could be a virus, a bacteria, parasite, something like that, okay? The first one that's going to end up taking place is the cell-mediated immunity. In a little while, we're going to talk about primary response and secondary response, and those are going to tie into these different immunity pathways. So cell-mediated immunity is going to activate phagocytes with the innate defense. It's also going to activate T cells. These cells are going to directly attack the pathogen cells and destroy them, okay? While this is going on and we're building up a recognition that we're seeing these, we start to shift gears and we begin producing B cells that create antibodies ultimately for the particular pathogen, so like chickenpox, okay? Then what ends up happening is the circulating antibodies bind with the pathogenic cell and either destroy it outright or hinder it in such a way that macrophages can come around and destroy them. Similar to if you watch the Storybots episode that I linked in the first video, they go into that process also, and they do a really good job explaining that one. So if you want a quick sort of run through how that works, I say go back to my first video and click the link for that Storybots, or just look for Storybots, How I Catch a Cold, I think is what it was called. But ultimately, both of these processes lead to the same thing, which is the destruction of the pathogens, the antigen-presenting cells, okay? So with adaptive immunity, we have antigens, which are our chemical targets. These are our some like signals. They could be attached to pathogenic cells, in which case they're abnormal signal flags. They could be individual molecules floating around in your bloodstream that are only present if a active infection has taken place, something like that. As they're floating around, or as the lymphocyte comes in contact with them on a particular cell, the cell becomes activated, in which case it starts cloning itself in response to that antigen. And so this is the beginning of the immune response. So because we now have a lymphocyte that is going to respond to that particular antigen. And so the idea is if that antigen is present in your tissues or in your bloodstream, there is something going on, so we need a lot of that particular lymphocyte to fight that particular infection. Once the infection is brought under control, those excess clones basically are removed. They're broken down. They're recycled because they're not necessary anymore. So here's a flow chart kind of showing what is going on. So basically, innate immunity is genetically determined. We're all born with the same level, okay? Right out of the gate, we all have this same response. Adaptive immunity, you become exposed. So when you're born, you don't have immunity to these various diseases. Now, a lot of your natural immunity comes from your mother, especially during breastfeeding, because a lot of those antibodies are transmitted from the mother to the baby. So that's one of the reasons why breastfeeding is so important. We talk about that in greater detail when we do the reproductive system at the end of the school year. We've got some different types of acquired immunity as we're going through here. And so the first one is active immunity. And so basically what that one is, you are directly exposed to it. Now, naturally acquired, you get sick. You got chickenpox, right? So in my example, I acquired an immunity to chickenpox by being exposed to the active virus. So that's naturally acquired active immunity. Artificially acquired active immunity would be after administering the antigen to prevent the disease. Those are your vaccines, okay? So now, like when you were growing up, you got chickenpox vaccines, right? So that's artificially acquired active immunity. You were artificially introduced to deactivated or dead viral particles in order to develop this acquired immunity. Passive immunity is transferring antibodies from another source. So naturally acquired would be through breast milk, for example. The other one is artificially acquired passive immunity, and that would be like antibody therapy. Plasma infusions and stuff like that, where basically you're taking somebody else's antibodies and they're being put into you directly. A lot of people were doing COVID-19 treatments using plasma. My sister is a nurse and she works at a plasma clinic, and right now a lot of their customers are people who have previously been infected and have recovered from COVID-19. And so they're donating their plasma with those antibodies, so then they can take those antibodies, they can transfer them to somebody else. That is artificially acquired passive immunity. All right, let us look a little bit at how T cells function in creating immunity within humans. So T cells are activated when they come in contact with an antigen. Now, you have antigen-presenting cells. So again, like one of your cells is infected with the flu virus, okay? It's going to present particular antigens that are unique to that strain of the flu or to chickenpox or something like that. These are known as MHC proteins, major histocompatibility complex, that will bind to these antigens. So the MHC proteins are on the T cells, okay? Two classes of MHCs. There's class 1 and class 2. You just need to know that there's two classes. We're not going to go into really any detail about what the different classes do. What you do need to know, though, is you have to have evidence of both in order to activate your immune response through the T cells. That's known as co-stimulation. Basically, you have to have two inputs in order to start this process. That's the big takeaway that I want you to get from that. All right, so here we have going on the what the MHCs are doing and stuff like that. So basically, on all of our cells, we have the MH1 protein, the class 1 MHC protein. All right. Immune cells will actually attack abnormal MHC complexes. You have class 2 MHCs, which are found on the lymphocytes and the antigen-presenting cells. The antigen-presenting cells, popping these up, are going to help spread immune cell production throughout the rest of the body. It's basically kind of like a message that it's being carried around, that it comes in contact with other blood cells and stuff like that. And so with antigen recognition, inactivated T cells have these receptors, and so they're looking for these antigen-presenting cells, right? They bind to the MHC complexes, and if there is a match at two sites, then that's the co-stimulation, and so now the T cell becomes activated, which is what we have right here on this page. Once it is activated, once the T cell is activated, then the activated T cell will actually destroy the antigen-presenting cell, which is right there. And then this diagram right here is a description of how that happens. We've got perforins that destroy the plasma membrane. We've got cytokines that stimulate apoptosis, which is that programmed cell death, releasing those lysozymes within the cell that basically chew everything up from the inside. Lymphotoxin, which is another substance that's released that disrupts cell metabolism. So all of these things are released all at the same time by those T cells. And then they'll float around and they'll find another cell that is not... that's abnormal, basically. This is another good picture that you should take and put into your notes, by the way. All right, so once we activate our T cells, based on what is being activated, we'll get differentiation from our inactivated T cells into our different T cells. So we'll get the cytotoxic T cells. Those are the ones that do the actual killing. right, that have the porphyrins and the lymphotoxin and stuff like that. We will also at the same time have the memory T cells start to be produced. And so these are going to be the T cells that have that are looking for the specific MHC complexes on our antigen presenting cells these are the ones that are going to respond to the particular antigens, basically. These cells are already active, floating around, and so if they come in contact with the antigen again, they will immediately start producing cytotoxic T cells. We don't have to co-stimulate at this point to do the initial activation. Regulatory T cells will also be present in small numbers, but those numbers will increase in the presence of an infection. And those will basically keep your immune system from going too far, activating too much, right? Being too aggressive. This will make sure that your immune system stays within the responses that are there. If you have allergies, for example, you know kind of what it feels like to have an immune system that is overactive, especially if you've got severe food allergies and things like that. Your immune system has a tendency to just keep going, and it will actually start to attack itself and cause a lot of problems. So that's what the regulatory T cells do for us. Now, if it comes in contact with the class 2 MHC protein, we get the development of the helper T cells, which stimulate the immune response. So they're going to actually work antagonistic to the regulatory T cells. And then we can also have the memory T cells that are produced to remember what's going on. Okay, so cytokines, it's one of those chemicals, we've talked about these a lot, right? Our chemical signals that are released by other cells that trigger our immune response. There's 6 major types of cytokines. Now, we're not going to go into a whole bunch of detail about the different ones, but the only one that we've really talked about are the interferons. But the 6 major groups of cytokines, you need to know the list. You need to know about interferons, but that's about it. You need to know that about interleukins, interferons, tumor necrosis factors. These are cytokines that are responsible with the cancer cycle. Phagocyte activating chemicals, colony stimulating factors, and then miscellaneous cytokines. The functions of cytokines will stimulate T cell divisions to produce memory T cells and accelerate cytotoxic T cell growth. It also stimulates and attracts macrophages to the area and stimulates cytotoxic T cell activity and activates B cells. Speaking of B cells do our antibody mediated immunity. They make specific antibodies for specific antigens, and then those antibodies attack those antigens. There's lots of individual B cells, and each one has their own antibody molecule. So every single pathogen that you've ever come in contact with, there are B cells just for that pathogen, and so just for that antigen, and foreign particles count for that too. So so anything that you have come in contact with, any foreign body that has come into you, there is a B cell that responds to that. And so basically, you've got a couple of stages that the B cells go through. So normally they're floating around, they are inactive, they've got antibodies on them right here, and then they have their class 2 MHC proteins, right? So if they come in contact with an antigen, it's actually taken into the cell, processed, and then spit back out to the surface of the cell, bound to the MHC proteins. And now it's ready to be activated. It is sensitized, right? So it's primed, I guess you can think about it. It's charged, it's ready to go, but it can't fully activate and turn into a plasma cell quite yet. What has to happen is the antigen-bearing class 2 MHC complex has to come in contact with a helper T cell, right? So our sensitized B cell comes in contact with a helper T cell, which will then cause the cell to divide into either a memory B cell, which is similar to a memory T cell, or a plasma cell, which will actively produce antibodies. Okay? Memory B cells, these little ones down here, are like memory T cells that will respond to the next time you're exposed to that. And then basically, they don't have to go through the activation process. They're already ready to go. So once you come in contact with that antigen again, they immediately activate, turn into plasma cells, and start producing antibodies. So the time that it takes to mount an effective response is way shorter the second time around than it is the first time around. And I think we're getting into what all that means here in just a little bit. So here is a diagram of the antibodies. They look like these little Ys. You've got one ends of the Ys, those are the antigen binding sites where they're going to stick to the antigens. And then you have your constant or heavy chain, your constant segment or your with your light chains and your heavy chains. Basically, just they're Y-shaped. You've got antigen binding sites on the two arms, and then there's a sort of center like anchor piece, basically. At the binding sites, you create what is known as an antigen antibody complex. Antibodies can bind to at least two different antigens. And once we create this complex, we sensitize our B cells and we mount our antibody mediated immune response. So once we start producing our antibodies, they are going to precipitate and agglutinate, which basically means they're going to start to clump together to create these massive immune complexes. This is also going to trigger the activation of the complement system, attract and activate phagocytes, opsonize your antigens to increase phagocyte efficiency. It's going to stimulate inflammation. These are going to prevent bacteria and viruses from sticking. Bacterial and viral adhesion is those cells sticking to your cells, membrane to membrane, and allowing them to go into cells or inject genetic material in the case of viruses. This is a complete antigen or a partial antigen, creating your antigen antibody complex, basically. And so we end up filling up both ports, is the takeaway on that one. The body's responses to antigen exposure, the first time you're exposed, that's known as the primary response. The next time you're exposed, that is the secondary response. The secondary response is more extensive and prolonged. The memory cells are already primed. So what does that mean? Basically, your body's response the second time around is more intense. You produce more cells. It's longer, so they stay active in your system for longer than the primary response, is what that means. Here's a graph showing us antibody levels in the primary response. IgM and IgG are different types of antibodies. I'm not going to have y'all go into all the different types of antibodies. But peak antibody levels hit at about 2 weeks after exposure to the antigen, okay? And then they drop off pretty quick after that. The secondary response is where we have activated memory B cells, and they activate at lower concentrations than the original B cells because they're already sensitized, okay? Because the primary response, there has to be a certain threshold in which something is wrong, basically, before your body notices and it mounts a response, okay? Secondary response, your body is, for all intents and purposes, it's actively looking for something. So it's kind of like if you're walking around and something bad happens to you and you never prepared for it. You never thought about it. You never thought that this would happen to you. But then after it happens, right? A lot of our safety responses, seat belts, helmets, safety protocols for fire drills and hazmats and active shooters and things like that, those are all done in response to events that took place. We know what we know about fire drills, for example, for hazmat situations, because of events that have happened in the past, right? And so now we're more aware of those things, we're monitoring, we're looking, so our response time is better. It's basically what's happening with secondary response. And the other difference is the actual response, once it happens, is overwhelmingly larger than in your primary response, okay? We don't just produce a small amount of antibodies. We just dump our body full of antibodies. And then the antibodies stay in our system for a significantly longer period of time, several more weeks. The graph, it actually goes off the scale of the graph. So you've got antibodies for these particular pathogens for 6 weeks, 8 weeks, or even more beyond what the primary response was. And so basically what this means is your body is, like I said, your body is looking. And when it sees something, it is like massive retaliation, overwhelming force. If there's a fire, firefighters don't just pour a little bit of water until it stops. They completely soak the area. And then once everything's saturated, they add more water to it until there is absolutely no doubt. And they have completely flooded everything. But it is impossible for another fire to start. Okay, and that's basically what your body does. It's scorched earth. We're going to try to kill a fly with a bomb sort of thing, right? That's your secondary response. Now, coincidentally, your second time you get exposed to a pathogen, you will never know it. Your primary response is when we start to develop the symptoms, right? The symptoms is that pathogen getting into your body and being able to be active and act on your cells and infect those cells and things like that. So those symptoms that you experience is the pathogen being allowed to work and reaching those certain thresholds, and then, hitting the fever and the inflammation and all that other stuff, right? So your primary response is when you get sick. If you get exposed again, you hit secondary response, and it is so quick and so overwhelming, you don't manifest symptoms. Or if you do, they're super duper mild, and you dismiss them, basically. Which then gets us into this idea of something called immunocompetence. Immunocompetence is your body's ability to mount a response after being exposed to the antigen. So your memory B cells and your memory T cells add to your immunocompetence, your ability to mount a response if you get exposed again. Let's see. Talking about bacterial infections, we've got combined responses, right? So neutrophils and NK cells begin killing the bacteria. Cytokines draw phagocytes to the area, and those phagocytes begin consuming cells and cell fragments and things like that, and sending out more chemical signals. Sending out those signals are going to activate helper T cells and cytotoxic T cells. The activation of those cells is going to trigger the activation of B cells and cause them to differentiate into plasma cells, which are going to then increase your antibody levels. And so in the primary response, that process takes more time. Okay, that's your cell-mediated immunity, typically, is in your primary response. Then it sort of shifts into antibody-mediated immunity and your antibody-mediated response with the production of the antibodies, and then it kind of goes back. So there's a sort of flow back and forth between cell-mediated and antibody-mediated immunity. And so this graph is just showing that there's different activation as we go over time. Neutrophils, that innate response, and the NK cells are going to activate first. They're going to activate fast. Then as we ramp up some of these other cell types, like the neutrophils and the natural killer cells, they back off. Then the macrophages are going to activate. While those are activating, the plasma cells are activating, right? And then about the time the macrophages are dropping off, the plasma cells are peaking, right? And then as all these other cells with the cell-mediated immunity are dropping off, well, here's our antibody levels, and our antibody-mediated immunity is peaking. And so they're sort of handing off that baton as they're going through, and something else more efficient, more effective, basically, the production starts to ramp up. Okay, so differences between viral infection and bacterial infection. The sequence of events are similar. The initial steps is where we start to get the differences. So cytotoxic T cells provide the adaptive defense. NK cells provide innate defense. So if we take a look at this real quick, we see cytotoxic T cells with bacterial infections because they're going to destroy the cells directly. We still have the activation of the helper T cells, which are going to lead to the B cells and ultimately to the antigen or the antibody production. Now, in the viruses, what we have to wait for is the antigen presentation by our own cells, because viruses work by injecting genetic material into our cells and hijacking them. Bacteria don't do that. They just directly start infecting cells. So we don't have to have our own antigen-presenting cells. The antigens present on the foreign bacterial cells, okay? But we end up with antigen presentation right here in the same place. We still end up with activation of helper Ts and B cells and antibody production and the destruction of the viruses. Now, the antibodies destroy the viruses directly, whereas in bacteria, the antibodies they will directly destroy the cells, but they also allow for phagocytosis by those macrophages and those other phagocytes when they form that antigen-antibody complex. Cytotoxic T cells are also going to activate. Those are going to directly destroy the infected cells. NK cells are going to destroy our infected cells instead of destroying the infection-causing cells like the bacteria. Okay, so there's a little bit of a difference on how they run. Copy these, put these in your notes and study them to kind of look at the subtle differences. And it has to do with the life cycles of the viruses versus the bacteria. Okay, the development of immunocompetence starts in the fetus with exposure to antibodies and pathogens via the mother and the placenta. This typically happens in about 3 to 4 months, so within starting the second trimester, basically. The fetal thymus cells migrate to tissues and become APCs, antigen-presenting cells. The liver and the bone marrow produce the B cells. So they're called B cells because they're produced in the bone marrow. The T cells are produced and finalized in the thymus, T for thymus, right? Let's see. Mama can confer immunity in utero, like I said, across the placenta, also by breast milk. This will eventually replace in utero immunity, and then their own exposure to pathogens after birth, either through direct exposure and you get sick, or through the use of vaccines, antibody transfusions, that sort of thing. All right, stress in the immune response. Stress actually can limit the immune response. Stress, typically when you're stressed, you secrete glucocorticoids that limits your immune response. Chronic stress can actually inhibit your immune response and lower your resistance to disease. So if you feel stressed, you have a lot of anxiety and stuff like that, you can actually you're more likely to get sick and you stay sicker longer. Glucocorticoids, we've kind of talked about this real quick. They reduce inflammation. So they can also be used medicinally for inflammation purposes. They reduce activity of phagocytes in peripheral tissues. They inhibit interleukin secretion. So in a nutshell, another reasons why chronic stress is bad for you, because it actually suppresses your immune system. All right, specific immune disorders. If you have allergies, those are known as hypersensitivities. Your body has decided that a relatively harmless foreign particle is considered invasive, and so your body reacts to that. So things like pollen, peanuts, stuff like that, it overreacts, basically. Autoimmune disorders like rheumatoid arthritis, for example, is one. Lupus is another one. Rheumatoid arthritis, basically your immune cells attack the tissues at your joints and destroy them and break them down. Lupus is another one that's an autoimmune disorder. Immunodeficiency diseases, these are diseases that basically remove immune cells. HIV is an immunodeficiency disease because it targets helper T cells, right? And so basically it removes that coordination step, it removes that cytotoxic T step, it removes that block, that chain from that entire process. So your whole, you don't activate B cells, you don't activate other T cells and stuff like that, because you remove those particular types of cells. Let's round things out a little bit. So what happens as you get older to your immune system? It starts to slow down. So your immune capacity diminishes as you get older. Right now, you don't really have much to worry about it. Once you get to my old age and exalted wizened status, like I've talked about in the past, and elderly, you're more likely to get sick. You're more likely to develop cancer because your immune surveillance is decreased and stuff like that. And part of that is because that thymus sort of, starting to atrophy and stuff like that. T cells become less responsive. You have fewer responsive T cells, which means you have fewer responsive B cells, and immuno surveillance also declines. So here we are. Here's this familiar diagram that we've looked at before. All of the different systems that interact with our immune system, right? Integumentary system is really critical for our innate defenses. Skeletal system, we've got cell production in bone marrow. Muscular system protects lymph nodes and lymph vessels and stuff like that. The nervous system coordinates the movement of a lot of this stuff and the production of different substances all through the endocrine system, the cardiovascular system, helps distribute the different immune cells and cytokines and things like that throughout the rest of the body. It also collects a lot of stuff that is drained into the lymphatic system. Well, let's take a deep breath, ladies and gentlemen, because that is it. This one's a little bit longer than some of the other ones, but we've got some complicated stuff going on. Now, I would recommend go back, study those diagrams that I highlighted in this video and that are also in the notes slide. Follow those flow charts. There's some really good explanations in the textbook if you go to the study area about a lot of these processes. I definitely think you should go through. Crash Course has some good stuff. Storybots, like I said, I mean, it's made for kids, but they do a really, really good job hitting all the stuff that I did, and let's be honest, in a more entertaining and easier to understand way than I could ever hope to do, because those little robots are amazing. So study those. Keep the different cells straight and the different processes straight and stuff like that. Those processes, that's going to be the big thing that you're going to be leaning on when you are making your project. I'm not really going to go into any detail right now. I'm going to wait until launch day. But make sure you have those immune processes straight, because that's going to help you come launch day when we get into our projects. If you have any questions, let me know. Send me an email, send me a remind message, ask me in class, drop a comment down there. I'll get back to you as soon as I can. Otherwise, I look forward to seeing y'all again soon.